Welcome back, class 10, to the online bio classes. We had already started the third chapter, genetics, and in the previous class you had already studied about the different types of or the traits and the different terms which are used in genetics, like alleles or allelomorphs, homozygous, heterozygous, dominant, recessive characters, and so on. Now today we will continue with the laws of genetics, which was law of dominance given by Mendel. Mendel, is known as father of genetics, and he gave three laws. So what are the laws of genetics? Law of dominance. Okay. This is the first law. Mendel gave three laws regarding genetics based on his monohybrid, that is a cross in which a single character was considered, is called monohybrid cross experiment. Okay. So law of dominance, what does it state? You should understand these three laws nicely. Law of dominance states that in the F1 hybrid, out of a pair of contrasting character, I am not writing, I will just explain. In the F1 hybrid, out of a pair of contrasting characters, like this one is pair of contrasting characters, only one is able to express itself, the other one remains hidden. The character which is expressed is called dominant character, and the one which is not expressed is known as recessive character. Okay? So law of dominance states that in a monohybrid cross experiment, means such experiment in which a single character is considered, in the F1 generation only one of the character is expressed, either tallness or dwarfness. So here tallness is expressed, that is known as dominant character, and the recessive character is the dwarfness, which is not expressed in F1 generation. Okay. So once again, what is law of dominance? Law of dominance states that out of a pair of contrasting characters, only one is able to express itself in the F1 generation. The character which is expressed is called dominant character, and the one which is not expressed is known as recessive character. Now the second one, that is law of, the second law is known as law of segregation. The word segregation means separation. This law is also known as law of purity of gametes. So according to this second law, that is law of segregation, at the time of gamete formation, like when the gametes are formed, both the alleles, they separate, like this capital T and small t. This is one of the parents having this type of gamete. Capital T, small t, it is called heterozygous. So it contains alleles for dominance, that is tallness also, alleles for dwarfness also. Both of them, they remain present together without mixing or blending with each other. Means neither the tall and the dwarf characters will mix to form a medium size height of the plant, nor they will mix up with each other. And at the time of gamete formation, when the gametes will be formed, both of them will separate. So this is called law of separation or segregation. Why it is called law of purity of gametes? Law of purity of gametes means, although both the alleles of two different characters remains together, they never get mixed up with each other. And at the time of gamete formation, they simply separate or segregate from each other. So this indicates that the gametes are pure for their characters. Okay. Now we will study about the dihybrid cross. Next one. What is dihybrid cross? As the name indicates, dihybrid means, di means two. Hybrid means in which two characters are considered. So cross in which two characters are considered is called dihybrid cross. I am not writing, I will just explain. Dihybrid cross means such a cross. Cross means what? In which we take two different plants as parents and we help them to bring about pollination and fertilization by selfing them or by crossing them. So dihybrid cross is such a cross in which two characters are considered. A cross in which two characters are taken into account is called dihybrid cross. Now I'll give you one example also to show the process of dihybrid cross. One example is round and yellow seed. A plant having round and yellow seed, each crossed with, they are the parent, P means parent here. A parent plant having round and yellow seed, each crossed with another P plant which has wrinkled. If it is round here, the recessive character will be wrinkled, and if it is yellow, the other character will be green. So round and yellow, two characters are there. One is shape of the seed, round and wrinkled. This is the shape of the seed. The other one is the color, yellow or green. So since two characters are considered in the plant while doing the experiment or crossing, this type of cross is called dihybrid cross. So round and yellow seed, plant having round and yellow seed, each crossed with a plant with wrinkled and green seed. Now we'll write down their genotype, or what are the alleles which are present in round. So I will denote, or we'll write it with capital R, capital R. Capital R denotes the round seed, and capital Y denotes the yellow color of the seed. It is crossed with wrinkled. So whenever we write, we use alphabet. Okay. So if you are taking capital alphabet for any character, like round, capital R you have taken. For wrinkled, you will not take W. Okay, you have to take R only, but it should be the small letter, that is a small r, small r. Here you have taken yellow as capital Y because it starts with Y, the yellow spelling. So here you cannot take G as green. No, you have to take this capital, instead of capital Y, you have to write the small letter Y. So I have denoted like this as a small letter and capital like this. Okay, this one is clear. So what is this? They are the constitution or the genotype of the organism, of both the plants. That capital R, these are the genes present, capital Y, here small r, small y, small y. Okay. Now we'll write the gamete formation. This is important to know how the gametes are formed. Okay. So from here, the gametes which will be formed is like how to form the gametes. You just learn it. You write one capital R. Okay, and then one small r. Now see, two I have taken. Now if you take this one, you take one capital Y and here small y. Here also you take one capital Y and one small y. So gametes which will be formed is capital R, capital Y. First one, this and this I have combined. Then capital R, this and this I will combine, a small y. Then third one, a small r, okay, and capital Y, then a small r and a small y. Here you can see both are small letter, means they are the recessive character. Here small r, capital Y is there, here capital R, small y is there. So just check the spellings. Capital R, capital Y, capital R, small y, okay, and small r, capital Y. Now these are the four gametes which are formed. Here also all the four gametes will be same because it is same like this only. So what are the four gametes formed? By combination of the genes of both of them. From here what I have selected? One capital R, one capital Y. One will come, and from here what will come? A small r and a small y. This one is there. Now using these four, 1, 2, 3, 4, capital R, small, capital Y, small r, small y, if you combine them in different ways, so I have done like this only, capital R, small r I have written. If you want, you can take here capital Y also. So if you take capital Y here, I will just change it and show it, then also it will be same, it will be correct only. Okay, if you have any doubt. Now see, first I combine from here, both are similar, capital. So from this one pair I have taken one capital R. From this capital Y, one pair I have taken one capital Y here. It is crossed with here also one pair small r is there, so one small r I have taken. and small y, one pair is there, so one small y I have taken. Now, how to form the gametes? Capital R and capital Y. This one you can take. Okay. Now you can, just like I had done earlier, small r. If you do like this, now check, it will be coming wrong. Why? Because if you write here small r, capital R, small r, such genetic constitution is not possible. Okay. So the one which I had selected, that one is correct. Okay. Now I will show you the combination. It's not possible to do here the Punnett square. So I will try to draw the Punnett square to show the dihybrid cross. Okay. You will find that total 16 plants will be produced. So let's check what are they. Capital R, small y, small r, capital Y. Then the third one was capital R is small, capital Y is small r, capital Y. Then capital R is small y, and the last one was both small. Okay. Here also same will be there. This one if you take as the gametes of parents like father, then this one mother gametes, capital R small y, here small r, capital Y, then capital R small y, small r, small y. Okay. Now we'll just make a box to combine all the gametes. Okay. This is the first row, then the second row, then the third one, and the last one. Okay. Now let's combine them. This one capital R, here also capital R, so write both capital R together first, then both are small y, so write both the small y together. Okay. Capital R, capital Y is also there. No, this one is wrong. Capital Y should be there. One capital Y should be there. Okay. Yeah. So here capital Y, capital Y. Now it's okay. Capital R, it should not be similar. Capital R, capital Y, small r, capital Y, capital R, small y, small r, small y. Now it's okay. Now second one, in the same way we have to fill all. Capital R, small y, capital Y, both. Okay. Then here capital R, just fill it up and then see what the result is there. Capital Y, small y. Here also same. Capital R, small r, capital Y, small r, capital Y. Okay. Then capital is small, then capital is small. Here small, one capital, one small. Okay. Then next column, both capital R, then capital Y, small y. Here also capital R, capital Y, small y. Then next one capital R, then small y. Then here it is capital R, small r, and both are small y. Now last column we have to fill. Capital R, small, capital small. Here small, capital small. Then here we have to do capital R, small r, both small y, and the last one is both small only. Okay. Okay. Now let's see what are these characters. What do they indicate? All right. First one is round and yellow. Now let's see how many of them. We'll try to get the phenotypic ratio. Phenotypic means physically how the plants appear. Okay. So this one is round and yellow. I am writing here round, yellow. The plants having round and yellow seeds. Here also round and yellow. So I will just tick mark the round and yellow one. Although it is small r, but since capital R is dominant, so it will show the character of round and yellow. This one also round and yellow. Tick mark I have done means same, this character only. Round and yellow, all tick mark I have done. Okay. Here also round and yellow. Here also round and yellow. Here round and yellow only. No, sorry. This is not round. This is wrinkled. So I write the character name so that you can identify. It is wrinkled and yellow only. Because both are capital Y, so it is wrinkled and yellow. So this one I will cross it like so that you can understand. Now this is round and yellow. So tick mark I am doing. This one is wrinkled and yellow. Because capital Y, small y there. So I will cross it here. So 1 cross, 2 cross, okay, to identify them. Now in the next column you can see round and yellow, so tick mark. Here also round and yellow, tick mark. Round and it is green. So now different characters coming. Round yellow, wrinkle yellow. Now this one is coming as round and green. This one is different, so I will write it here. Now this one is round and yellow, round and green. Round and green already we had done. Round and green is not done. Yeah, round and green. So this one same only. So here also round and green. So two are same, round and green, round and green. Now next one, round and yellow. This is round, this is yellow, no? So round and yellow means tick mark. Here it is wrinkled and yellow. Now see, this is also different. Wrinkled, wrinkled yellow. Here, cross only, yeah. Wrinkled and yellow. Okay, cross you can do. This one is round and green. This one only, round and green. Round. How to know round and green? You may be surprised. How sir is saying this one is round and green, this one is wrinkled and green. Just by looking at this, capital R, small r, capital Y, small y, small y, small y. If both one is capital R, one is small r, it means the character is round. Why? Because one is capital R, and that is the dominant character. But if both small y there, means it is green character. If capital Y, that is yellow character. If one capital Y is also there, that is yellow character. And the last one is wrinkled, because both are small r, and green. Now let's check the ratio. That is called phenotypic ratio. How many are round and yellow? Let's count it. 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 is 2. I am writing here, okay? Hopeless, so I will just write it. I will rub these dates and all. Okay, what is this ratio I am writing? This is called phenotypic ratio of dihybrid cross. It is asked in the exam also, this ratio, phenotypic ratio of dihybrid cross. Of dihybrid cross. Okay. So what is first 9 I have done, means this one is completed. Now next one is wrinkled and yellow. How many wrinkled and yellow is there? Let's check once. I have shown it with the help of cross mark. So wrinkled and yellow, 1 is there, here 2 is there. Any more cross? Yeah, here. So 3. So 9 is to 3, these 2. This is also done now. This is also done. Now which character is left? This one. Round and green. Let's see how many round and green are there. 1, 2, 3. So 3 are there. So next ratio is 3 is to wrinkled and green. Only this one is there. The last one is wrinkled and green, because all the alphabets are small. Small r, small r, small y, small y. So it is 1 only. So this is the ratio of phenotypic ratio of dihybrid cross. Understood? Let's check it once and try to draw it yourself so that you can learn more about it. Okay? Okay, now first two laws already you have done, and the dihybrid cross also, and you have seen that what is the ratio of phenotypic ratio of dihybrid cross. It is 9 is to 3 is to 3 is to 1. If you want to get to know about its genotypic ratio, it's quite lengthy. It is 12 is to 12 is to 24 is to 12 is to 1. So it will be quite long. Okay, that is not required in your syllabus also, but I will just say what is the phenotypic ratio. 12 is to 12 is to 42 is to 12 is to 1. So that is not needed. Now next, what is the third law of Mendel, that is known as law of independent assortment. Law of independent assortment. Okay. Law of independent assortment. Okay. Now, what does this law says? Law of independent assortment, which is the third law of Mendel. It states that in a dihybrid cross, dihybrid individual, dihybrid means having two characters. So if an individual has two characters, the segregation means the separation of the genes or the traits of one character. So the law states that when two pairs of traits are combined in a hybrid, segregation of one pair of characters is independent of the other. So segregation of one pair of characters is independent of others in a hybrid. So hybrid means having two different characters. So in a hybrid, when two pairs of traits are combined here, the separation of those traits is independent. Like if suppose I will give the example, just now we had done the dihybrid cross in which round and yellow seed, these two different traits were there, round and yellow. Now separation of this round and yellow seed, like round can combine with, it was done with wrinkled and green, wrinkled and green. Okay, so two traits are there in both the parents, round yellow and wrinkled green. Now whether the round seed will be with yellow color or it will be with green color, it is independent. So seed can be round, it may be yellow in color or green in color. So if round like this, if yellow, capital Y, or it can be round or green. So it's independent that whether capital Y or small y, which character for which particular trait will combine with which parents. So separation or the segregation of one pair of character, one pair means here two pairs of characters are there. So this pair will combine with yellow color or it will remain with green color, it is independent. Okay, so learn the definition of that law of segregation or law of independent assortment. Now next one we have to do is, after this, after the Mendel's law, first law, second law and third law of genetics, the next topic which is there in our syllabus is tongue rolling. What is tongue rolling? I will say it is a character in which a person is able to roll his tongue. So very few people are able to roll their tongue, like they will make a tunnel shape, others cannot roll. Okay, so those person who can roll, they are called the tongue rollers. Those who cannot, they are called the non-rollers. Similarly, the other character is the earlobe. This is the earlobe. Now earlobe may be attached or it can be free. Free means the hanging one. So attached earlobe or free earlobe. Both different types of earlobes are there. Now next is the next topic which is there is sex determination in man. Sex determination in man. Okay, you know that human beings have 46 chromosomes. How many? 46 chromosomes are there. That is 23 pairs of chromosomes are there in each and every cell. Now out of these 23 pairs of chromosomes, 22 pairs of chromosomes is called autosomes. They are similar in both male and female. And the last one pair of chromosome is called the sex chromosome, which consists of X and Y chromosome. They are called the sex chromosomes. So in case of male, what is the genetic constitution or what is the chromosome number? What type of chromosome is there? 22 pairs of autosomes. A I am writing is autosomes, plus if it is a male child, the chromosomes will be XY. So 22 pairs means 44 plus 1, 45 plus 1, 46. Understood? This is 22 pairs. If you don't want to write 22 pairs, then you can write 44 plus XY. This is the male child composition. 44, 45, 46. So normal chromosome number is 46 in the human beings. Similarly, in the female child, the chromosome is, or the chromosome constitution is 22 pairs, same like this only. Autosomes are same because they are similar in male and female. And the last pair of chromosome, that is 6 chromosomes, is XX. So we can write it as 44 plus X. So this is the female child composition. Okay. Now the organisms which produce eggs, they are the female, and those which produce the sperms are called the male. Inheritance follows the law of segregation. In man, sex chromosomes or sex is determined by the chromosomes. If chromosomes are XY, then the male child is there. If it is XX chromosomes, it is homozygous. Same chromosomes are there, XX. That determines the chromosome number of or the chromosome of females, not of males. Now, sex determination by chromosomes. It was proposed by McClung. Okay, name of the scientist. In most plants and animals, it is determined by the allogenes and the sex chromosomes only. Now, the female has 2 X chromosomes. Here you can see male has only 1 X chromosome, but male has this Y chromosome which is not present in females. So, the male chromosomes, the X body or X chromosomes, this X chromosomes was discovered by a scientist. His name is Henking, and the Y chromosome was discovered by Stevens. Okay. Now next is sex-linked inheritance. So you should know what is the meaning of sex-linked inheritance. Apart from this certain body characters which are attached or linked with X and Y chromosomes, they are called the sex-linked characters. And when these characters are transferred from the parents to their children, such type of inheritance is called sex-linked inheritance. Okay. Now some examples are there. Diandric sex-linked or X-linked traits means certain characters are there which are associated only with the X chromosomes. We will study now, like hemophilia and color blindness. But some characters are there which are associated only with the Y chromosomes, like the presence of hairs in the ear, and so it is dominant or it is found only in the males. Females do not possess such type of characters. Baldness is also there, which is present on the Y chromosome. It's not found on the X chromosome. So what is X-linked chromosomes? X-linked chromosomes means those chromosomes which are attached with X chromosomes and passed from the parents to their children. And 2 examples which are there that you have to study is hemophilia and color blindness. Okay? Hemophilia and color blindness. Okay, look here now. The first X-linked character I have written, that is color blindness. It was discovered by the scientist named Wilson in 1911. Now, what type of disease is this color blindness? It is a disease in which a person who is affected by color blindness with these defective genes, he is unable to distinguish between 2 colors. What are they? Red and green color. Red and green color he cannot distinguish. So if he is given a composition of red and green color, he may not be able to distinguish which one is red, which one is green, or distinguish between them. So 2 types of names are there. Protanopia. Protanopia means red blindness, but if a person is unable to see the green color blindness is there. If red color blindness is there, it is called protanopia. If green color blindness is there, it is called deuteranopia. And this is caused by recessive gene. What is recessive gene? The genes which are not showing their characters or expressing themselves in the first generation, but they remain present in the individual. It can show the character in the next generation, and it is common in man. So mainly the man is suffering from this type of disease. So very rare in females. The women are not suffering from it, very rare. And it follow criss-cross inheritance. It's important to understand what is the meaning of criss-cross inheritance. Criss-cross inheritance means, like if this one is father, okay, from father it goes to the daughter, and then from the daughter to the grandson. From father to the daughter, not going to the son. So here I have written father to son, no. So this character will never pass from the father to the son, but instead of that, it will pass to the daughter and then from the daughter to the son. So that type of inheritance in which the characters pass from the father to the grandson through the daughter, not through the son. Okay? So this is about color blindness. Now next we have to study is one example I will show you about color blindness. Okay, now let's see one example of color blindness I am going to show. First one is the parents. What type of parents are there? Here P represents parent. One parent is a normal mother. So I'll just write it and then their genes. Normal mother and crossed with a father who is color blind. Color blind father. Let's write the genotype now. Normal mother means you know XX is there. Okay, and here XX means normal mother, not suffering from the defective character. So instead of writing like this XX, I write like this. Okay, it's crossed with, just to mark the cross here. Color blind father, it is XY. And where is this defective gene present? It is present on X chromosome and it is represented by a small c. So a small c I have written means this is the defective gene. Okay, then this one, if you want in the book it is shown capital C, I am not showing that one. Capital C means it's normal. This small c recessive allele is shown here. This capital one is shown here in the book. So this one means normal, not suffering from disease. This is capital, this is small, means this is the recessive character. And color blindness is a recessive disease, recessive character linked disease. Okay, and it is associated with X chromosome only, not with the Y chromosome. So what type of gametes will it produce? I'll just leave it so that it will not make you confused. So what are the gametes produced? Let's see. Combination or first gametes we have to make. So gametes are this one X, X, and here also two gametes will be there. One X, small c, and one Y. Okay. Now the F1 generation. Let's see what are the combinations of F1 generation. This one and this one, that is X, X, C. Means this is a girl child who is having or who is getting a defective allele of color blindness. The second one is XY, that is a normal daughter having no defective allele. Third one, this X and this one, she is also getting a defective allele, means suffering from color blindness. And the last one, this is a normal male child, not suffering from the disease. So this one is about F1 generation. Now let's see what happens in the F2 generation. If this one, the son-in-law, this XX is crossed with the carrier female, this one is the carrier female, XXC. Now what will happen? Let's see what happens now. Now here XX means the daughter, sorry, we have to make it male here, no? XY. This is the normal male and it is crossed with the carrier female. Normal male is there. Here the male was color blind. Now here the male is normal and crossed with, the normal male is crossed with color blind female. So what are the, like this is the parent generation, the gametes will be XY. Here the gametes will be X and XC. That is in the F2 generation. This one was F1, now F2 generation is coming. This one and this one, if you combine, XX, that is normal female, this one, and first one, yeah, wait. Then first one and second one, this one, XXC, that is the defective allele is present. Now second one and second one, that is XY, and second one, XXC. So what you will see that normal female, color blind female, normal male, color blind female. Okay, so here also you can see that the son should be color blind here. Yeah, XC, Y, it should be Y here, last one. Wait, I'll make it clear because it's too much confusing here. The space is not there to draw. Okay, so XY, normal male crossed with the carrier female, that we have to show. Okay. Look here. Now normal male crossed with carrier female. This one, this is the parent generation if I take. Then their gametes, XY, here the gametes, XC and X. Then in the F2 generation, there was no place to draw, now it is clear. So first one, that is X and XC, that is she is the defective, but she is not suffering from color blindness. Why? because only one of the recessive allele is there, no, XC. In both it is not there. No, wait. Then this one and second one, that is XX, normal female. Then this one, first one, XCY, that is the defective male. XY. So here you can see that there are four, in which the normal son is there. This son is suffering from color blindness. So son is color blind. In the previous one we saw that the daughter was color blind. So this one came from where? This one came from the daughter. The character for color blindness, it came from the defective allele of daughter. This is the daughter's chromosome. So it is passed from daughter to the grandson. So this type of inheritance is called. Now earlier in the beginning when I had drawn, it was the color blind male. Then from the male it came to the female. Okay, and then from the female it came to the male once again. That is, it passed to the grandson through the daughter. So such type of inheritance is called criss-cross inheritance. Hope you understood this criss-cross inheritance. Okay, now next one we have to do is hemophilia. What is hemophilia? It is also known as bleeder's disease, in which a person suffering from this disease lacks certain clotting factors which are required for the blood to clot, so that it will prevent loss of blood from the body. Okay, and that is anti-hemophilic globulin. A protein is there which is absent. In normal person, the clotting can occur within 2 to 8 minutes, but a person suffering from hemophilia can bleed till death, and it can be very serious. It is also a sex-linked recessive character, and since this character is present in the royal family of England, so this disease is also called the royal disease. Okay, like Queen Elizabeth and all, they are suffering. Queen Victoria is suffering from this particular disease, and the genes are located in X chromosome. So Y chromosome has no such inheritance. So I will show you one example of this one also. That is the example of hemophilia. Okay. Okay. Now look here. The second one, that is hemophilia. I have shown. Parent, father and mother. This father is normal, and here the female I have shown a small h. This h represents the hemophilic genes which are located on X chromosome. Okay. Now it means this female is suffering from hemophilia. Alright. So in the what are the gametes formed? XY, here X and XH. In F1 generation you will find this normal female is there. See the carrier, because he has only one defective H. Okay. And here the male is normal, but here the male, that is child, grandson, he is suffering from hemophilia because the defective gene or the H is located here. So in the first generation, the daughter is carrier, but the son is suffering from hemophilia. Now in the F2 generation, if I take these two as the parents, that is XXH and XHY, means the son is suffering from, this parent is suffering from colorblind hemophilia, and the female is only a carrier. Okay, because she has only one H, defective allele. So you can find, in order to make the female suffer from the disease, both the X chromosomes must contain the defective allele of H. Like here you can see the gametes are X and XH. Here the gametes are XH and Y. Now if I combine them in the F2 generation, X and XH. So it means she is the carrier of, she is a female child and she is carrier of hemophilia. Then X and this one Y, this is normal son, XY is normal grandson, not suffering from hemophilia. Now if I take the second character XH and XH, means she is the female child, XX chromosome is there, and both chromosome has the defective genes. It means she is suffering from the disease. I have made a circle. Then XH and Y means this is a grandson, and grandson is also having the defective genes or alleles. So he is also suffering from hemophilia. So in the second generation or F2 generation, we find one of the grandson and the granddaughter, they are suffering from hemophilia. But in F1 generation, it was only the grandson who was suffering from hemophilia. Whereas the female was suffering, she was only the carrier. Carrier means the one who contained defective alleles but not suffering from disease. Okay, so with this we have completed this chapter number 3. Now you can try and solve some of the questions which are given at the end of your book. Like I will just read out some previous year board questions. Name the following. Practice this type of question as homework. A pair of genes responsible for a particular characteristics in an individual. 2007. The answer is, it is called alleles. Now, transmit characteristics from parents to offspring. Who transmit the characters from parents to offspring? The answer is, it is the DNA which is present, or the chromosomes also you can write, they transmit characteristics. The suppressed alleles of a gene, it is called recessive allele. Okay. The physical expression of genes in an individual, physical expression is called phenotype, and the genetic constitution is called genotype. Similarly, differentiate between genotype and phenotype, definition you have to write. Then some diagram based questions are there. We'll practice it here, 1 or 2 questions, so that you can understand. Okay. Thank you. And right here, once you watch the video, just in the comment section you write your name so that we can come to know how many of you have seen the video. Okay. Thank you.